Welcome back to the lecture on CRISPR. And in today's class, we will have a quick review of whatever we learned, and then we will see how it can be designed or modified for application. So in the last class, let's get through the simple ones. CRISPR first, clustered, regularly interspersed short palindromic repeats. And then we also learned about another protein, a protein called as Cas, CRISPR associated protein. And then we also have one more sequence that is called as protospacer adjacent motif, that's called as PAM sequence. So CRISPR Cas, these are several genes that are present, several genetic elements. Some of them code for sequences like this. There are repeats, a spacer, repeat, a spacer, repeat, spacer, like this you will have. If, in a way, when you study the CRISPR array, okay, if you study the CRISPR array properly, we might probably learn that a bacterium is there, and every time there is a kind of infection, and there is, if it is in a location A, for example, and there is a kind of phage here, and that phage will infect, and probably it will, some of the sequences will get incorporated into the genome, right? Say, for example, this bacterium has moved on to a different location where there are different types of another type of phage, say phage C, just for sad. So from the earlier infection, we got this bacterium got the phage DNA from here, and from the later infection, it will incorporate another sequence. Like that, you can, if you properly look into the spacers that are present here, okay, you can probably record the kind of history. Using bioinformatics tools, you can say the ones that are on this side are the most recent ones, right? But again, bacterium will be infected by, there are at least 10 times more of bacteriophages than there are bacteria apparently. And there are many types of bacteriophages too. So if you keep it like this sequence will keep on growing, right, within the genome. But what happens is these sequences that are present in the CRISPR region on this edge, okay, if it is the genome is extended, these will usually eventually vanish, or they will get selected away, meaning that once they are no longer infected, these have no function anymore, right? They will be going away. So the repeats that you see here is a kind of history of whatever wars these bacteria have won against the phages. That's a nice way of also looking at it. And they're also associated with Cas9, Cas proteins. So there are several steps in that. Adaptation, we learned about it, how Cas1 and Cas2 proteins pick up DNA from the invading phages and then incorporate them into the host DNA, right? And then we have expression and maturation, where the RNA is produced, which will form hairpin loops. This hairpin loop is because of the palindromic nature of the repeat region. And what you have here is the spacer. And then by processing RNase, we will get CRISPR RNA. CRISPR RNA will combine with that of Cas proteins and form a ribonucleoprotein, which now is kind of acquired immunity. When any phage infects, it will bind here and have further cleavages. And that is how the host bacterium is saved from the viruses. I hope you remember some of the things that we have already discussed. And then we also have naive acquisition. The first time a protospacer is picked up from the foreign DNA, we get a spacer. And next time, using this, this is the spacer, new spacer that is made. Using this spacer, the CRISPR can cleave further more of the DNA, because of which we will form more of these protospacers which can be incorporated into the CRISPR genetic elements there. So we also discussed about how CRISPR sh CRISPR should be able to distinguish between self and non-self, right? Because the same sequences are present in the foreign DNA and also now upon integration also in as spacers within the genome. So somehow the genome should be protected. Otherwise, causing double-strand breaks there would have lethal effects many a times. So if the difference is in the self DNA, and at least in this part, if you look at it, it is clear enough. Where in the with the self DNA, the genome part, there is repeat, there is a spacer, and another repeat. And the CRISPR RNA that is produced will have these regions also base pairing. Whereas the foreign DNA, the DNA that is not self, will have different sequences here. Only the target would actually match, right? So these will be degraded, but the self DNA will not be degraded. So that is how self and non-self will be distinguished. And these are the two important things, right? They should have memory. Memory is present. It is based on the spacer. And it is spacer, when we say it is based on the nucleotide sequence. And there is a mechanism to distinguish between self and non-self. It is also based on base pairing. It is based on how many base pairing and to what extent, right? Based on that, the Cas9 activity is there, and then cleavage of the target sequence would happen. And then we discussed about a little bit about Cas9. There are several domains in that. Some of these important domains are that of the PAM interacting sequence. A PAM sequence is required, and for the activity of Cas9, there are HNH and RUVC and REC2 and REC1. They are bridging, and it's a lot. It's a complex protein. Yes. So we also discussed this part. What happens, and we need actually two RNA molecules. One of it is called the tracer RNA, the tracer RNA, and the CRISPR repeat that we will get from here, CRISPR RNA, and we'll have Cas9 protein from here. That is what will make sequence specific cleavage, right? You need this whole ribonuclear protein. You see this maturation tracer RNA, CRISPR RNA, and Cas9. The three together will form a complex and mediate the cleavage. Yes. And here we have RUVC and HNH that will make the that will make the cleavages, the subunits of Cas9 protein. Okay, so what we are going to do now is people have identified these genetic elements. They are very interesting. We can use them because they are nucleases. First thing is they are useful in genetic engineering because we can cut them. Not just any random cut should be made, because say for example several endonucleases are there, exonucleases. They are not sequence specific. In that, if they are not sequence specific, they are not useful, because we want to be sure where to cut and and then only we can make appropriate recombinant molecules. We have a goal, right? Goal of making certain type of recombinant DNA. If we want to do that, we need to have specificity, right? There should be nuclease activity, there should be specificity, and we should have more control over them, their activity. Then we can make those things. So people have done many advancements, and the first advancement that we need to discuss is that they have mixed So what we need is tracer RNA and CRISPR RNA, and we need the Cas9 protein. So what people have done, because we don't have to add, now we need 3 components, tracer, CRISPR, and Cas9. So how can we make it into a 2-component one? It will be simple if we can fuse this using this linker loop. Now we only have 1 RNA, which is a single strand DNA, or it is a single guide RNA. That gives us more control instead of having 3 or 4 factors, trying to express them, keep them their quantity in appropriate numbers so that they will form a ribonucleoprotein. Here we fused it, so we are always sure that we have appropriate amount of both these things, and it is a lot more simple. So the typical CRISPR-Cas9 system that people use for genetic engineering, editing, or any of those things is to have a single guide RNA or guide RNA, and then Cas9 in an expression vector. So we can have this. All you need to now change is this target sequence, right? And then we can always choose. Say, for example, we have a DNA, and there is a unique sequence here, and here. We can change this target sequence there so that we can produce a CRISPR RNA that is specific to this. And then, of course, it should come as a ribonucleoprotein along with the Cas9, and that will cleave this DNA at this certain location. But one thing we need to remember is Cas9 has a PAM binding motif. I'll just call it as PAM motif for domain. The activity of Cas9 is dependent on the interaction of PAM sequence with that of this protein. Okay, if there is no PAM sequence, then the Cas9 cannot cleave the target DNA. There are different types of PAM sequences, and they differ from one organism to another organism. In some cases, like most often what we will see is Streptococcus pyogenes, that is there, it has NGG. In a different organism, you may have CCN and TCN and so on. And some of them could be much more complex. And this is simple enough though. So if a Cas9 should work on a DNA, there are 2 requirements that we need to have in the target sequence. First thing is it should have this PAM sequence. Say here we will say nucleotide N represents any nucleotide and GG. And what else we need is we need to have a sequence, CAGT. I'm just giving a random sequence here. Only when we have such an arrangement like this, and if we have made a CRISPR RNA that is having that, which has this sequence here like this, say, and I'm just maybe putting it reverse, but that's all right. Just understand the concept. I'm just trying to put what is the significance of PAM sequence. Say, for example, you had some sequence like this, okay? And there is also similar sequence somewhere else. CAGTAGT. The difference between sequence 1 here and site 2 here is that one of it has the PAM sequence, NGG, whereas the other one does not have it. If we use the same, if the CRISPR RNA were to come and bind here, assume, because we said we have designed a sequence like this, it will not cleave here. That does not get cleaved. Why is that? There is no PAM sequence here, because of which the activity of Cas9 is not, it's not activated, because of which it cannot cleave it. Whereas in the site 1, there is PAM sequence, and there is also this sequence where the base pairing would have occurred, and then Cas9 would cleave the site. Okay, I hope you understood the significance of the PAM sequence. So now we can plan some things. So if I had a gene, if there was a gene, and I'm trying to put up the limitations, okay, and I want to cleave exactly here and here, then the problems associated with that would be the problems associated with that would be or requirements is that, say this is a whole genome and we have a gene here, and I want to, say for example, cut that gene. Okay, cut the gene in these locations. If I want to do that, I need to design on the target there should have been PAM sequences here and here. And if there are PAM sequences, then I can design CRISPR RNA for this specific site here. Say I'm just putting it like that. Okay. If there are no PAM sequences exactly here, then the CRISPR RNA would not work. But as I showed you in the previous slide, there are many types of different Cas9 proteins are available and from different organisms. And probably with more research, I think we will find more of these. I'm unsure how effective they are. But the important thing is we may get some variety here or some choice in the future. So that is one thing you need to put in mind, that there should be a PAM sequence you should have identified, and then you should have designed a sequence, CRISPR, that is complementary to the target site. One another complication that we need to keep in mind is, if I'm cutting a genome here, and I've chosen that way, we should ensure that there are no other sites that are specific to the same CRISPR RNA. Say if we had a site here, the one is having a PAM sequence followed by the spacer, complementary sequence to the spacer. If similar site is somewhere else, that part will also get cut, right? PAM sequence followed by complementary region sequence similar to that of the targeted region. Those are called as off targets. They are unintended. What we want is to make a genetic change cut here and here, for example. But if there are sequences similar to somewhere else, they would get cut here also. We do not want those things. This I'm talking especially in terms of genome editing, right? In genome editing, when we are talking about transgenic animals, for example, I discussed this. We want to change the genome, and we want to make changes only in one place. As I hope I will try to repeat this statement. We want to have change. We want to have intended change, okay, and avoid unintended changes. Unintended changes or inadvertent changes. That means we should be sure that the process that we are doing in one location here to the DNA or the genome should not have induced any kind of genetic change anywhere else in the genome. That is a very important thing, especially if we are looking for any changes that we want to do for clinical purposes, medical purposes. So we have to be very careful about this important thing. And even now, when people say genetic engineering, and if you say the public can get a little bit worried about what will happen, that it will change many things. It might change even transgenic animals or transgenic plants even. We have a lot of trouble trying to bring them to the market. There are a lot of regulations, because when we do any kind of change here, we have to ensure that there are no ill effects or unintended changes. and the intended change should be highly calculated and should have been tested to rigorously before implementing it in anywhere else. So there are a lot of websites or online tools. If you give a sequence, and then the online tools will figure out the um, best spacers that you can use. Okay, they run algorithms to figure out algorithms, and if you say the organism name, everything, these online tools will give you a list of spacers based on where you want to cut and the availability of the PAM sequences. And the third point is, and prevent off targets, right? We do not want any off targets. So these are some of the criteria the algorithms will use. This one is where to cut. You can specify the sequence. I want to cut in this particular gene. Say I want to make a cut here like this. Then anywhere from here, I want to cut this gene X. Then the program, if you give the sequence and tell the organism name and other things, it will figure out and calculate which is the best spacer that you could use, and that will not be present anywhere adjacent to a PAM sequence. Okay. Now let's see how what happens when we are trying to do genome editing. So we assume that we have designed CRISPR sequence, and then based on we designed a single guide RNA and Cas9. We expressed them. They would have recognized a particular site and they have cut it. What would happen to it? One of it is will undergo non-homologous end joining. Okay, that means these two, the edges, the ends will be processed a little bit. Some bases may be removed, and then the repair mechanisms will bring these two ends together and join. So there is some disruption in the genetic information. There could be insertions or deletions based on the degree with which the repair enzymes work on how the enzymes process these ends. Mostly they are deletions. So if there is a gene I have taken and we have cut it here, and it is now in two pieces, and then these were processed, these edges were processed, so some parts might be lost, and then they would be sealed together to now this is a shorter one, and most likely its reading frame is affected, right? There will be a frame shift mutation kind of thing. So this particular gene, even if it is expressed, it will not be able to produce a functional protein. So we have deleted a gene, right? At least we deleted a gene function. Yes, it's a simple way to do that. And that's why usually double-strand breaks are very toxic. We need, in eukaryotes, we have very dedicated systems, repair systems to join, and that is usually not involving homologous recombination, so we call it as non-homologous end joining. There is another way of doing things. That is, say we have cut the DNA. The DNA is cut. Okay. Now the DNA CRISPR Cas using the single guide nucleotides, single guide RNA, the DNA is cut. What should happen is there is another way of how repair can happen. That is by, if we have provided a DNA with something like this, DNA molecule, and I'm providing something else to it. I'm providing a little bit of additional DNA like this. The blue ones that I have drawn here are homologous to this region here. And this region I would have designed homologous to this region. So we have homologous regions. And now homologous recombination, if it occurs, what would happen? You will get the red, the gene that I'm drawing as red, will get inserted into it. Now we have done gene insertion, right? We have replaced. We made a cut, provided a provided a gene of interest. That's not the bothering. For homologous recombination to happen, we have added these homologous regions, because of which homologous recombination would have occurred. Along with that, the gene of interest would get inserted, right? We can do these things. It's not so if there is a genome, we can delete, we can insert in the genome very accurately. One of the reasons for this ability is we'll also have the PAM, the spacer sequence could be somewhere around 15 to 20 bases. It depends on the system, of course. And because of that, we have much more specificity, right? If you remember, if there are 4 bases, it is about 256. Probability of its occurrence is every 256 base space. If it is 6, 4096 or something like that, right? 4092. If it is, you can calculate how much would it be, the probability of its occurrence. If it is something like 20 bases, it's very, very rare. And not only that, this particular 20 sequence should be adjacent to a PAM sequence. That is another level. So it's likely that it's very specific, but there will always be some off targets. Say, for example, I have chosen 20 bases. At the target site, it is perfect base pairing. But at some other sites, maybe out of 20, it is base pairing to 18, and there too, the Cas9 may cut. So those are off targets that we have to keep in mind. So is it just about always cutting, or can we do anything else? What they have found, or they have engineered, are some of the mutations. In PAM, in Cas9 protein, they have done Cas9. There are two regions, RUVC and HNH, which are responsible for making the double-strand cuts like this. So they have made a mutation, which is D10A nickase. So aspartic acid is replaced with alanine. Aspartic acid is an acidic amino acid. Alanine is a non-polar amino acid, so it doesn't have the active groups. So the RUVC is the function of RUVC is lost, or the ability to ability to cut one of the strands is lost. So now we have a Cas9 protein that only makes single-strand DNA cuts. There are also other mutations where they have made histidine at position number 840 replaced with alanine, because of that mutation, the activity of HNH is lost. So it also makes a single strand cuts, but on the opposite strand of the PAM sequence. Okay. So people have done something else. They introduced both the mutations. Well, let me see this. So we can use these strands in differently, where you can have single strand cuts on both these sides like this. Okay. Double Cas9 nickase is, you're using two different spacers. You have single guide RNA. This is one single guide RNA, and this is another single guide RNA. What I mean here is there are two different sequences here, right? We're using Cas9, that is with a mutation that doesn't have the RUVC nickase activity, but we are using two different, so that, say, for example, I want to cut, this is the gene, say, and I want to make a cut here, and I also want to make a cut here. So what I missed in the other points is, if you want to make these cuts and you have designed the CRISPR, say which points here, as well as like this, you can decide what spacer. All you need is, of course, the PAM sequence should have been there. You can decide the same thing on the other strand as well, on the other side of it. And then you are deciding, right? This spacer here is different from the spacer that you would have used here. And then we would have Cas9 would cut here as well as here. And then we have cuts there. That is how you can use on both the sides. I forgot to mention in the previous slide. Anyway, so it gives us a lot more flexibility of how we can use those strands to make single strand breaks. We can also make another activity. 1 second, let me zoom out. So here is a protein, okay? And if you put in both the mutations, D10A and H840A, this Cas9 doesn't have any activity at all, any nickase activity. It can recognize the single guide RNA, it can recognize the spacer, everything, but it does not have the nickase activity or the cleavage activity, because of which it doesn't make any DNA strand breaks. What is the useful use of that? We still have a lot of use for it too. I will also I will repeat it so that you will probably understand the mechanism. Whenever we are trying to use a tool, I would rather like that you try to understand the molecular mechanisms thoroughly. So I hope you are able to see these are the images from a different one. These are better illustrations. So, and using multiple illustrations is always better. So here is the sgRNA, which is a chimera of tracer and CRISPR RNA. It has this hairpin loop, which comes from the repeats. And the green one shown here is the spacer sequence, sequence that is specific to the target site. And here we have the PAM sequence, which is NGG. So the nuclease Cas9 would have cleaved it on both the sides like that. And what is the consequence of it? There could be, you could have two sequences. First one, here in this case, we only made one single strand break. What can we do with that? How can we delete the gene? We could delete it by we are actually interrupting or disrupting the gene, not deleting the gene. In that case, there are two possibilities: non-homologous end joining. We just allow it to heal, and because there will be insertions or deletions, the gene could be inactivated. In other cases, we provide a donor DNA with homologous regions. So homologous recombination may happen, because of which insertion may happen. This is called as homology directed repair. Then there is another way that we just discussed a little while ago. Say I want to get rid of this whole region. How can I do that? We can have one sgRNA prepared for target A, another sgRNA prepared for target B. And now if you allow it, a cut will be made here and also here. And then you're deleting the whole gene, unlike in the first one. In this case, we are deleting this whole fragment. What we will now have is whatever DNA we'll keep. Or we can also probably allow it to undergo end joining, and this region could undergo end join with the other side. Okay. Now we can also have, I discussed about Cas9 that can have no nickase activity at all. Normally it would make a double strand break, but there could be mutations in 10th position as well as 840 position, because of which the Cas9 can perform every activity except cleaving the DNA. So what is the use of it? I want to know, say for example, there is a genome. This is the DNA, assume that. And I want to know where exactly is this particular sequence. Say I want to know where this gene is within the, say, assume it's bacterial, of course, circular I have drawn. So where is it present within the cell? Is there a specific location that it is present? If I want to do that, I can design the guide RNA to a location like this, specific to that particular where the gene is present. And how would I know where it is bound, where the guide RNA is present? It can only be said if we have the Cas9, which is targeted, which is coupled, translationally fused with the repressor, with a reporter gene, right? Then I know where in the chromosome, where in the cell this of the where in the cell this this gene is present within, when it is in the nucleus or in the bacterial cell, preferably nucleus. Say I want to do a different experiment now. I want to have this particular gene expressed. What can I do? All we need is the sgRNA, Cas9 translationally fused with an activator. If you put in activator, it will bring in other proteins of transcriptional machinery, remodeling complexes, and other things, and allow the transcription to happen of this gene. So, what did I do by doing this? I am able to express a particular gene. How did I do that? By putting in an activator. Why did the activator come here? Because the activator is translationally fused with the Cas9 protein. Why is the Cas9 protein here? That is because we designed a spacer that is upstream, right upstream of the gene that we want to express. So if we can express, we can also repress, right? All we need to do is make a translational fusion of Cas9 along with the repressor like this. So I hope you realize the power of it. You can inactivate a gene, you can delete the entire gene, you can put in a new gene, like in this case. We can also activate gene expression, we can repress gene expression, we can also visualize where in the cell is this particular gene. Visualization is also present, can be done, right? That is why CRISPR is one of the most powerful techniques in the recent past, and Nobel Prize was given for that in the last year, I think. These will be redundant, and I hope you will be able to understand it as it is there. This is the standard CRISPR-Cas9, makes double-strand breaks. Here we have a Cas9 mutant, which can make nicks on one side like this, single-strand ones, nickase. We can also have a single-strand inactive Cas9 mutant, which can go to the target site along with this guide RNA, but cannot do anything else. It can only perform other activities. I mean, based on what kind of translational fusions we have made. So these are just representations of other ones. How can I silence a gene? Right? Silencing a gene is it's an epigenetic modification where we can methylate the DNA. This is a good one. RUC minus and HNH minus, in the sense their activity is lost. So if we give a spacer and a single guide RNA like this, and if it were translationally fused to a DNA methylase, or a domain to which the DNA methylase would bind, then this whole DNA will be methylated. And if this DNA is methylated, it cannot be, often it will not be expressed at all, right? That is how we can silence the DNA as well, some of the genes. Okay, I hope you understood the concept of the CRISPR RNA. If you have any questions, I would like to take it now. Please let me know if you have any questions. Anybody? No questions? Okay. So I will stop the recording, and maybe before that, I would want you to remember a I gave you an assignment on Google Classroom. I hope you have noticed it. I wanted you to make questions, and these questions are for 20 marks. This is how the pattern will also be for your exams. We will discuss about it in later, or maybe I might have discussed. Part A, you have to write 4 out of 6 questions, and the marks will be 4 into 20 is 80 marks. In part B, there will not be any choice. You will have one question into 20 marks. That is total of 100 marks. Okay. So the activity or the assignment that I've given is for you to make some questions. The idea behind it is that when we know how to make questions, and firstly, the idea is that questions can be based on memory. Of course, everything is kind of memory in a way, but say, um, what is this? Is a memory. I can show you something and ask, what is it? And the other ones would be analytical. This particular course is about, I mean, both are, well, let me say it again. So, memory for me is more of knowledge. Analytical and application, if you have, that is for me intelligence. You may disagree, but and just at least for the sake of this discussion. So when we make questions, majority of the time since our childhood, we will be asked more about memory and less of analytical ones. Maybe mathematics and others will require a lot of analytical thinking and others. In when it comes to biology, mostly it is about memory. But our course, rDNA in medicine, is a analytical question, analytical subject about how to analyze, how to design, how to apply. It's an applied science, and for that we need to have a lot of intelligence, or we need to practice that applying thing, and that is how we improve our intelligence. So when you make a question, I would want you to make more questions based on analytical. You can look at the whole syllabus. You can look at videos. You can try to emulate what is there in the videos. But I want you to make questions that are more of analytical side. Each question is 20 marks. It can have sub questions, 2 sub questions, A and B, for example. So one of it you can make memory, and the other part analytical. Okay, so the best questions whoever sets will get a gift from me. And the idea about this whole exercise is that if you can think like a teacher, right, like the question paper setter, and if you are able to make good quality questions, then you know how to make, you must have been already knowing the answers, especially these ones, analytical ones. If you're making a question, you must have known either known the answer or how to figure out the answer. And either way, it is a good exercise for all. And imagine if all you 20 or 30 people made these 4 questions. Okay, I need 4 questions for each one from each unit. If you made, and we made a database of questions, and we circulated it to everybody, we will have fantastic revision as well. And we can probably also discuss some of the questions that people may not have understood. And we know who has submitted which question, and we can always ask them for help. I hope you will take it seriously and try to do your best, and try to make analytical questions as much as possible. Okay? Yes. I'll stop here. If you have any questions, you can ask.